So we're going to look at Hardy-Weinberg equilibrium in the next couple of videos. And the really most important thing to remember in Hardy-Weinberg equilibrium is that when we are in this HWE, there is no evolution. No evolution. So what does that mean? That means that there is no change in allele frequency. Now, usually when we're doing a Hardy-Weinberg problem, we're just looking at a single genetic locus. So we're going to say that there's no change in allele frequency at that one gene, at that one spot in the genome. And then we say there was no evolution. But we actually can test this using the Hardy-Weinberg equations. We can test to see if evolution's happening because our equations, our expected and predicted, will not end up being the same thing. We'll see some differences, discrepancies. So whatever we observe in terms of either allele frequencies, sorry, that's spelled wrong, or genotype frequencies. Let me go back and fix this. I needed to pick the object eraser. So whatever we observe, that's still wrong. For allele or genotype frequencies, we're going to compare it to what we expect or what we predict in terms of genotype or allele frequencies. Now notice that I wrote those upside down from each other because when we compare these, we're going to usually be comparing our observed allele frequencies to expected genotype frequencies or our observed genotype frequencies to expected allele frequencies. We actually use one to predict the other, and then we do our comparison. So I'll do an example problem of this later on for you. Okay, so observed, we're going to use that to predict something, and that prediction is going to be our expected value. Okay, whatever we predicted is what we now expect from our observations. That's the whole idea behind this Hardy-Weinberg equilibrium, is to compare our observed and our expected and see if there are discrepancies. If they're the same or very close to the same, and for that we can use something like the chi-squared test. If they're the same or close to the same, then we are in Hardy-Weinberg equilibrium and there were no allele frequency changes. All right, so let's think about an example. We're going to look at cats, and that is because cats are the one animal I can draw with any kind of consistency. So here's our little population of cats, and these cats are going to have a couple of different features. We're just going to look at one different feature, so one feature with variation, and I'm trying to copy paste it here, but it always takes me a second to do it. It probably would have been faster to just draw a whole new cat. Look at this, there. If we'll stay up, I can paste it. Like now I have two cats, they're so cute. All right, so some of our cats are gonna have tufted ears. Some of our cats are not gonna have tufted ears. These are the little tufts. I personally think these tufts are adorable, like what lynxes have. Some regular, like feral cats or house cats have these too. So some have tufted ears and some don't, and we're also gonna have an intermediate phenotype that I'm gonna do a close-up on that have little teeny tiny tufts. All right, so we're going to say that our cats with tufted ears, we're going to say that they have got a specific genotype, and they are going to be big A, big A. And our untufted ears will be little a, little a. Now, this is an additive trait, an additive genetic trait, and that means that our heterozygotes are going to be intermediate between our two homozygotes. So this isn't just a regular dominant recessive type trait here. This is an additive trait. So let me draw in our heterozygotes as big A, little a, and I'll just do a close-up on their ears here, which have these intermediate phenotypes, these short little tufts instead of the big fancy tufts that we see in our big A, big A's. So if you have 1A, you've got fancy tufts. If you've got, if you've got both big A's, you've got fancy tufts. If you've got both little A's, you've got no tufts. And if you're in between big A, little a, then you've got these intermediate type of tufts. So in our Hardy-Weinberg equilibrium with these little cats, we expect to see the same frequencies of cats with big tufts, medium tufts, and no tufts at all every single generation. So I want to talk about our big letters that we're going to come up with again and again in Hardy-Weinberg, which are P and Q. So P and Q are just representing frequencies. So P is going to be the frequency of one allele and Q will be the frequency of the other. Now there's only two. They're always going to have to add up to one, and that's because there's only two alleles. So if you take all of the big A alleles and add it up with all of the little A alleles, you're going to have to add up to the number 1. So our P is the frequency of the big A allele. Q is the frequency of the little A allele. So just like flipping a coin, when you flip it, it's always going to be heads or tails. If you flip it 100 times, you add up the heads and tails, you equal to 100% of your flips. So that's what our 1 means. Our 1 means 100%. When we add up frequencies, we should add up to 100. So what is a frequency? A frequency is just the number of the observed over the number of the total, the number of your particular category over the number of your total. So let me just write that out here. So our number of A alleles over the number of total alleles. So number of A plus little a alleles. Okay, just like your number of heads over your number of heads plus tails, that's your frequency of heads in a coin flip. Okay, so number of little a alleles is the number of, the frequency of little a alleles is your number of little a alleles over your number of big A plus your number of little a, your total number of alleles here. And what we want to see is if these P and Q change over time. And the way that we're going to calculate that is by looking at the Hardy-Weinberg equations and looking to see if we're able to predict the next generation, okay, by looking at the first generation's P and Q values. So are we going to get back out the same as what we put in, or was there selection on one or two of these? Now, what's important to know is that this part in the blue box, it will not change. This is always true, whether or not you're in Hardy-Weinberg. These are the equations we use to make our predictions, but your P and Q are always going to add up to 1. It doesn't matter if you're in Hardy-Weinberg or not. P and Q will always add up to 1 if you're in a system with only 2 alleles at a particular locus. So don't think that just because P and Q added up together equal 1, that means you're in Hardy-Weinberg. It just means that you know how to add 2 numbers together. Again, this is always true whether or not you're in Hardy-Weinberg equilibrium. So again, the way that you're going to tell is by comparing your observed to your expected. So you'll either start out with your allele frequencies or your genotype frequencies, and you're going to use those to calculate the other end. So if you start out with allele frequencies, you'll calculate your predicted genotype frequencies and see if they actually match what you observe for genotype frequencies.